Okay, so today we're going deep on something pretty fundamental, to life itself.
Yeah, right.
Water.
Water.
If you think back to like intro bio, your body is mostly water, like 70% water, give or take.
It's wild, right?
Yeah. And it's crazy to think like even when you look at like a solid object or, like pick your favorite celebrity like Elon Musk or something. Like
Yeah.
at the cellular level, even he is mostly water.
Yeah, all that cytoplasm, all those organelles floating around in there.
Exactly. Mostly water.
So today we're talking all about water,
Mhm.
and specifically for you guys in Mr. Martin's IB biology class, we're diving deep on this concept of water potential.
Yes.
And more importantly, what drives water to move into and out of cells.
Right.
So we're going to unpack all of these key ideas, and hopefully in a really clear and simple way, especially if English isn't your first language.
Yeah, we've got some great resources for today's deep dive, including materials specifically for IB biology, SL, and HL students.
Absolutely.
Plus, we've got some extra notes to really break things down step by step. Yeah, so by the end of this, you should have a really good grasp of what influences water movement across cell membranes and how plant and animal cells handle this kind of movement differently.
Exactly.
So to get started, let's just look at water itself.
All right, so let's start with the basics. The water molecule.
Okay.
H2O,
H2O.
2 hydrogens and an oxygen,
Right.
joined by a covalent bond,
Covalent bond.
sharing electrons.
Okay, so they're sharing electrons.
Yeah, but that sharing isn't exactly equal.
Uh-huh.
Imagine it's like a tug-of-war for those electrons.
Okay.
Oxygen's a bit stronger, pulls the electrons a bit closer.
So oxygen is winning the tug of war.
It is. It's got a strong grip on those electrons, and because electrons are negatively charged,
Right.
that means the oxygen end of the water molecule ends up with a slight negative charge.
Okay.
And because the hydrogens are having their electrons pulled away, they get a slight positive charge.
Right, so the hydrogens become slightly positive.
Exactly, and that's what we mean when we say water is polar.
Polar.
It has these slightly charged ends.
Okay, one side's negative, the oxygen side, and then the hydrogen sides are slightly positive.
That's it.
Got it. And so this whole polarity thing, this leads to hydrogen bonds, right?
Yeah, exactly, because you've got this slightly positive hydrogen on one water molecule.
Yeah.
It's attracted to the slightly negative oxygen of a nearby water molecule.
Right.
And that attraction is a hydrogen bond.
So it's like an intermolecular force?
Exactly. It's weaker than those covalent bonds within the H2O molecule itself.
Right.
But like our notes point out, you've got so many of these weaker bonds going on at the same time,
Uh-huh.
and that's what gives water some of its unique properties.
So all these water molecules are like interacting with each other through these hydrogen bonds, and they're constantly forming and breaking.
Yeah,
Which is why water is liquid at room temperature.
exactly.
Okay, so this polarity, these hydrogen bonds, this all makes water a great solvent.
Absolutely a solvent. That's what something dissolves in to make a solution.
Right, the solvent dissolves the solute.
Right, and because water can dissolve so many different things.
Yeah, it's like the universal solvent.
Right, exactly. When water is a solvent, we call the mixture an aqueous solution.
Aqueous solution.
So think about dissolving sugar in water.
Okay.
Water is the solvent, the sugar is the solute, and then the sugary water.
Right.
That's the aqueous solution.
Okay.
And the process of it dissolving, we call that solvation.
Solvation.
So the solute gets solvated by the solvent.
Got it. Okay, so we've all heard that phrase like dissolves.
Right.
So what does that actually mean in practice?
So basically polar substances,
Okay.
which are also called hydrophilic,
Hydrophilic.
which means water-loving.
Water loving.
They like to dissolve in polar solvents, like water,
Makes sense.
whereas non-polar substances
Okay.
are hydrophobic.
Which means
Water-fearing.
water-fearing, they don't dissolve so well in water. Okay, so similar things like to stick together.
Exactly.
So can you give us an example of a polar substance dissolving in water?
Sure. So let's think about salt.
Okay, sodium chloride.
Sodium chloride's NaCl.
NaCl?
It's an ionic compound
Okay.
made up of charged particles. You've got your sodium ions, which are positive, and chloride ions, which are negative.
Okay.
And when you put salt in water, the water molecules, they're attracted to those charges.
Uh-huh.
So the slightly negative oxygen ends of the water molecules,
Right.
they surround the positive sodium ions,
Okay.
and then the slightly positive hydrogen ends,
Right.
they surround those negative chloride ions.
Okay.
So you can kind of picture it as these hydration shells forming around each Ion.
Hydration shell.
Yeah, they basically separate the ions and allow the salt to dissolve.
So it's like the water molecules are clustering around each ion and pulling it from the others.
Exactly, and that's an example of ion dipole forces at work
Ion dipole forces.
between the ions and the polar water molecules.
Got it. Okay, so what about other polar substances that aren't ionic?
Right, so for polar molecules like glucose,
Oh.
which has lots of OH groups
Uh-huh.
that are partially charged, a similar thing happens. Those water molecules are attracted to those partially charged areas on the glucose molecule through what we call dipole forces.
Dipole.
Yeah, and they can even form hydrogen bonds directly with the glucose molecules.
Okay.
And so all of that allows the glucose to spread out and dissolve in the water.
Okay, so anything with charges or partial charges is more likely to interact with water.
Yeah, exactly.
Now, what about the hydrophobic substances?
Okay.
Like fats and oils that we know don't mix with water.
Right. So as you said, those are nonpolar molecules.
Yeah.
They don't have those significant partial charges that water is attracted to.
Okay.
The water molecules are much more attracted to each other, through their hydrogen bonds
Right.
than they are to those nonpolar molecules.
Yeah.
So the nonpolar molecules end up getting pushed together, minimizing their contact with water.
Which is why you see like oil droplets forming in water.
Exactly. They're just trying to get away from those water molecules.
They don't want to hang out. All right, so water's ability to dissolve stuff really comes down to its polarity and how it interacts with these charged or partially charged molecules.
Precise.
So now let's switch gears and talk about how water moves across cell membranes. This is where things start to get really interesting for biology.
Absolutely. The movement of water into and out of cells is so important for their survival.
Right.
But there's a bit of a hurdle there.
What's the challenge?
Well, a main part of the cell membrane,
Yeah.
it's made up of those phospholipids.
Right.
Remember those fatty acid tails? That hydrophobic core?
Hydrophobic.
Yeah, and water being polar.
Yeah.
It doesn't really like to pass through that oily layer very easily on its own.
So it's kind of like we were just talking about. It's like mixing oil and water. They just naturally want to stay separate.
Yeah, it's not a very favorable situation.
So how does water get across the cell membrane efficiently then?
All right, so this is where aquaporins come in.
Aquaporins.
Yeah.
Okay.
These are special protein channels embedded in the cell membrane. And they create a hydrophilic or water-loving pathway
Okay.
that allows water molecules to just zoom right through the membrane much quicker than they could if they had to go through that hydrophobic interior.
So they're like little water-specific doorways?
Yeah, basically.
Aquaporins, the cell's dedicated water channels. Got it. So with these channels present, then what is osmosis?
So osmosis is the net movement of water molecules
Okay.
across a semi-permeable membrane, like a cell membrane with aquaporins, from an area where there's a lower concentration of solutes to an area where there's a higher concentration of solids.
Okay, so the water moves to where there's more stuff dissolved.
Exactly.
Okay, and so what are the key things that need to be in place for osmosis to happen?
All right, so you need three main things. First, you need that semi-permeable membrane,
Okay.
like we talked about, often facilitated by aquaporins.
Right.
Second, you need a difference in the concentration of solutes across that membrane.
Okay, a concentration gradient.
Exactly. Water will move from the side with less solute to the side with more solute.
Right.
And third, osmosis is a passive process.
Passive.
It doesn't require the cell to use any energy or ATP.
Okay.
It just happens naturally, driven by that tendency for things to even out.
To reach equilibrium.
Exactly.
Now, you mentioned water potential earlier. Can you explain that a bit more in simpler terms?
Sure. So think of water potential as like the water's eagerness to move
Okay.
from one place to another.
Okay.
So pure water in an open container that's got the highest water potential.
Okay.
But when you dissolve something in water, it lowers the water potential.
Okay.
The water molecules are now interacting with those solute particles, and they're less free to move around.
Got it.
And pressure can also affect water potential.
Okay.
Think about squeezing a wet sponge.
Right.
It increases the water potential of the water inside because you're putting it under pressure.
Makes sense.
So water always moves from an area of higher water potential to an area of lower water potential.
So a solution with a lot of dissolved stuff has a lower water potential than pure water, and water will tend to move towards it.
That's right. This concept is explored in a lot more detail in HL biology.
Okay, so water potential is like the driving force behind osmosis.
Yeah, you could think of it that way.
So we've talked about osmosis. Now, let's talk about diffusion just to clarify the difference between the two, because they sometimes get mixed up.
Absolutely. So diffusion, that's the movement of any type of particle,
Okay.
a solute,
Uh-huh.
from an area of high concentration to an area of low concentration.
Moving down the concentration gradient.
Exactly, down the concentration gradient.
Yeah.
But osmosis,
Yeah.
that's specifically the movement of water across a semi-permeable membrane
Okay.
from an area of low solute concentration to an area of high solute concentration.
So diffusion is about the dissolved particles moving, and osmosis is about the water moving.
That's the key difference.
Okay.
Another difference is that diffusion can happen whether or not there's a membrane present.
Okay.
But osmosis, by definition, always involves a semi-permeable membrane, one that the solute can't easily pass through.
Right, because if the membrane was permeable to the solute would just diffuse down its concentration gradient,
Exactly.
and then you wouldn't see this net movement of water.
Right, osmosis wouldn't be the main event.
Okay. That's a really clear way to explain it.
Good, I'm glad.
So now let's talk about what happens to cells when they're in solutions with different solute concentrations.
Okay.
Isotonic, hypotonic, and hypertonic.
Right.
These terms describe how the concentration outside the cell compares to the concentration inside.
Right, so let's start with an isotonic solution.
Okay.
Iso means the same.
The same.
So an isotonic solution has the same concentration of solutes as the inside of the cell.
Okay.
So in this situation, water molecules are still moving across the cell membrane.
Uh-huh.
But the rate of water moving in is equal to the rate of water moving out.
So it's balanced.
Yeah, there's no net change in the cell's volume.
Okay.
We call this a state of dynamic equilibrium.
Dynamic equilibrium.
Things are happening, but there's no overall change.
Got it. So isotonic is kind of a happy place for the cell.
Yeah, you could say that.
So what happens if the solution outside is different? Let's talk about a hypotonic solution.
Okay.
Hypo meaning less.
Less, yeah. A hypotonic solution has a lower concentration of solutes compared to the inside of the cell.
Okay.
So because there's a higher concentration of solutes inside, water is going to move by osmosis from that area of low solute concentration outside the cell to the area of high solute concentration inside the cell.
So water moves into the cell.
Exactly. And that causes the cell to swell.
Now, what's the risk for an animal cell in a hypotonic solution?
Well, animal cells don't have a rigid cell wall,
Right.
so they can swell up so much that they actually burst.
Burst.
Yeah, we call that cytolysis.
Cytolysis.
Yeah, it's like blowing up a balloon too much eventually.
It pops.
Exactly, it pops.
So how do some single-celled organisms that live in freshwater, which is hypotonic, avoid this?
That's a great question.
Yeah.
So organisms like paramecium, they have this really cool adaptation called a contractile vacuole.
Contractile vacuole.
Yeah, it's this specialized structure that collects all that excess water that enters the cell from osmosis, and then it actively pumps it back out.
So it's like a little water pump.
It is a tiny little pump preventing the cell from bursting.
That's pretty amazing.
It's a form of osmoregulation,
Osmoregulation.
actively maintaining a stable internal water balance.
So it's like constantly regulating.
Yeah, constantly working.
Okay, now what about the opposite? A hypertonic solution.
Okay. Hyper meaning more.
More, right?
Yeah.
So in a hypertonic solution, the concentration of solutes outside the cell is higher than inside.
Okay.
So water is going to move by osmosis from the area of lower solute concentration inside the cell to the area of higher solute concentration outside the cell.
So the water moves out.
Yeah, then this causes the cell to shrink.
And this shrinking in animal cells, this has a specific name, right?
It does. It's called crenation.
Crenation.
The cell basically shrivels up, which can really mess with its normal functions. Right. So both hypotonic and hypertonic environments can be bad news for animal cells.
Definitely.
Which is why maintaining that isotonic internal environment is so important.
Exactly.
So how do multicellular organisms like us manage this?
So we have these complex systems in place to regulate the solute concentration of our body fluids, keeping them isotonic to our cells. And a key player here is our kidneys.
The kidneys?
They're constantly working to maintain that correct balance of water and solutes in our blood plasma.
Okay, so our kidneys are always hard at work.
Always.
Now, plant cells, they're different because they have that rigid cell wall.
Right.
So how does water movement affect them in hypotonic and hypertonic solutions?
Okay, so the cell wall makes a big difference
Okay.
in a hypotonic environment.
Uh-huh.
Water still moves into the plant cell by osmosis.
Right.
The cytoplasm expands and pushes against the cell wall.
Okay.
And that pressure is called turgor pressure.
Turgor pressure, that's what keeps plants like upright and firm, right?
Exactly. The cell wall is strong enough to withstand that internal pressure, so the plant cell doesn't burst.
Okay.
In fact, that turgidity provides crucial structural support for the plant.
So it's actually a good thing.
Yeah, it needs that pressure.
So in a hypertonic environment, plant cells become firm.
Yep, nice and firm.
What happens in a hypertonic environment for a plant cell?
So in a hypertonic environment, water moves out of the plant cell because of that higher solute concentration outside.
Okay.
And that causes the cytoplasm to shrink,
Right.
and the plasma membrane pulls away from the cell wall.
So it detaches?
Yeah, and we call this plasmolysis.
Plasmolysis. Okay,
Yeah.
so the cell membrane separates from the wall. What does this do to the plant itself?
So this loss of turgor pressure,
Yeah,
it leads to wilting.
wilting.
The plant loses its stiffness and starts to droop because those cells aren't pushing against the cell walls anymore.
Okay.
And if the plasmolysis is really severe, the plant cells can actually die.
So while that cell wall prevents bursting in hypotonic conditions, it doesn't prevent shrinkage and wilting in hypertonic conditions.
Exactly.
So our notes mentioned a way to figure out the solute concentration inside plant tissues.
Yeah, so you can do this by basically taking pieces of plant tissue, like potato slices,
Okay.
and putting them in solutions with different solute concentrations. Some more concentrated, some less concentrated than what's inside the cells.
Okay.
And then you measure whether the mass or length of the tissue changes in each solution.
Okay.
And by doing that, you can figure out that isotonic point
Right.
where there's no net movement of water and no change in mass or length.
Got it. So it's like a little experiment you can do.
Yeah, it's a classic experiment.
And I remember something about standard deviation and standard error when you're analyzing the results of that experiment.
Right, so when you repeat the experiment multiple times, you get a set of data for each concentration.
Uh-huh.
Standard deviation tells you how spread out those data points are around the average.
Right.
And standard error, that gives you an idea of how accurate your sample average is compared to the true average for all the cells.
Okay.
And you can actually show this variability using error bars on your graphs, which helps you see how confident you are in your results and helps you pinpoint that isotonic concentration more reliably.
So it helps you make sure that your results are good.
Exactly. It adds that layer of statistical rigor.
That's really cool. Okay, so now let's talk about why isotonic solutions are important in medicine.
Okay, so isotonic solutions are super important in a lot of medical situations because they prevent damage to our cells.
Right.
You know, that damage that could be caused by too much water moving in or out.
Yeah.
Because remember, our animal cells don't have that protective cell wall.
Right. They're vulnerable.
Exactly.
Yeah.
So can you think of any examples where isotonic solutions are used?
Well, I know that when patients need IV fluids for rehydration, those fluids have to be isotonic to their blood plasma.
That's right, like normal saline,
Normal saline.
which is a 0.9% salt solution.
Yeah.
That has to be isotonic to prevent those red blood cells from either swelling and bursting or shrinking.
Yeah.
Another example is when organs are being prepped for transplant.
Oh, right.
They're often kept in cool isotonic solutions to keep those cells healthy until they can be transplanted.
Wow. So even an organ transplant.
Yeah, it's all about preserving those cells.
And I guess things like eye drops and contact lens solutions are also isotonic.
Exactly. They're formulated to be isotonic with your eye cells to prevent any irritation or damage.
So it's all about maintaining that delicate balance.
It is.
So we talked a lot about tonicity, but are there other adaptations that organisms have to prevent these water problems?
Definitely. So we already mentioned the paramecium.
That's little contractile vacuole.
Yeah, actively pumping out excess water in those hypotonic environments.
Mm-hmm.
And then more complex organisms like us, we have our kidneys, which play a much bigger role in regulating our overall water balance.
Okay.
So for example, if you get dehydrated, your kidneys are going to try to conserve water, produce less urine to prevent your cells from shrinking.
So your kidneys are like the master regulators.
They are.
It's really fascinating how these different organisms have evolved these clever ways to deal with water balance at the cellular level.
It really is. It shows how fundamental water is to all life.
So just to recap, we've talked about water's polarity, its solvent abilities, how osmosis drives water movement across those cell membranes.
Right.
The effects of isotonic, hypotonic, and hypertonic solutions on animal and plant cells. The medical importance of isotonic solutions.
Yep.
And some of the ways organisms regulate their internal water balance.
It's been a busy deep dive.
It has been a very thorough deep dive.
It has.
So understanding these principles, this is really foundational for your IB biology studies, guys, especially as you move into more complex topics.
Yeah, especially when you start looking at water potential.
Right.
And its role in all sorts of biological processes.
Exactly. So here's something to think about. How could disruptions in normal water balance at a cellular level contribute to different diseases in both plants and animals?
That's a good one.
And maybe you could think about some other examples of how organisms that live in very different environments maintain their internal water balance.
Yeah, lots to think about.
Definitely. And to Mr. Martin's IB biology class, we wish you all the best in your studies.
Keep exploring these fascinating concepts.
And to everyone who tuned in and found this helpful, especially if you're also studying IB biology, take a moment to like, subscribe, and leave a comment.
Yeah, we love to hear from you guys.
Definitely, your support helps us create more deep dives like this. So stay curious, everyone, and keep exploring the amazing world of biology.
See you next time.